Alright guys, so every living organism has homeostasis, and homeostasis means, we have learned already, maintaining the internal environment of the body. Now plants are not as advanced, as complex like us or other animals. They are simpler organisms. We know that plants, they are just simply made up of organs. They don't even have organ systems. If we look at a plant, here are roots. They are one organ. Stem, another organ. Over here there are branches. Another organ. Over here are leaves. Another organs. There can be flowers over here if it's a flowering plant. Another organ, right? So all these organs join together to make one organism, that is what? Plant, right? While in humans or animals, what do we say? Organ systems join together to make an organism, right? Like we have circulatory system, we have nervous system, we have excretory system, right? These are the few examples. We have skeletal system, muscular system. All these systems, they join together to make a complete organism. So they don't have that much complicated physiological processes. They don't have a nervous system at all, nothing, right? But still they show homeostasis. Now, you guys know that all the plants, they have leaves. And in the leaves, in the daylight or whenever the light is available, one process takes place, that is photosynthesis. Now, for the process of photosynthesis, we already know plants, they take up water from the soil and they take up carbon dioxide from the air. They join, these both join together in the presence of sunlight and chlorophyll, and you get glucose and oxygen which goes into the air, right? Now over here, plants, they need to maintain the concentration or higher concentration of carbon dioxide inside the leaves, right? So that they can perform or they can continue the process of photosynthesis during the daytime. They also need to maintain the water. Now remember here, we are talking about the plants which are mesophytes. Who will tell me what are mesophytes? Who remembers? Who remembers what are mesophytes? There are three types of plants: hydrophytes, mesophytes, xerophytes. The plants living on land. Xerophytes are also on land, but they are in desert. They have very limited quantity of water available, right? Hydrophytes are the plants that live in water. They have excessive amount of water available, right? While the mesophytes are the plants that have moderate quantity of water available, right? Means that water is not too much, water is not too less. But the water can become too less in them in certain conditions. Let's suppose that if there is low humidity over here, around the plant, low humidity means less number of water vapors in the air. You guys know that the water, it is transpired from the leaves through its stomata. And what is this process called? Transpiration, right? Now, if the humidity is low, inside the leaves of the plant's water concentration, I will use the word because it is in the air spaces. It is as gas particles, not as liquid. Understand? So inside the leaf, the water concentration is what? high. Outside water concentration is what? Low. Because of low humidity, so water will move out by transpiration. So water loss will happen, and if the humidity is low, it can be an excessive loss of water. But plant has moderate quantity of water available. It should preserve that water. It should preserve that water. So how the plant can preserve that water? So at this condition, when there is low humidity outside, the stomata, they close. Stomata, what they will do? They will close to prevent the loss of water. That is homeostasis. Another condition, that is high temperature. Now, I have a question over here. We say that the enzymes inside the animal's body, they work best at which temperature? 37 degrees. Don't say 40. Let me tell you, I told you this thing last year. We talked about that enzymes can work in vitro and in vivo. Now, who remembers what is in vitro and what is in vivo?
Inside and outside.
What is inside, what is outside?
Inside.
Now, out of this, which is inside and which is outside? Very good. In vitro, remember in vitro fertilization, IVF, that is outside the body of the living organism. So, in vitro is outside the cell, and in vivo means inside the cell. So inside the cell, human or any enzyme, the optimum temperature is 37 degrees. Outside the cell, it is 40 degrees. We know that, right? Who knows what is the optimum temperature of the enzymes in vivo for plants? 30 degrees. Right? So the optimum temperature for the plants is 30 degrees for the enzyme activity. If the temperature becomes higher, what will happen? Plant needs to cool its body down. Now what do you do when you are feeling hot? Sweat. And when you sweat and when the air brushes your body, there is a cooling effect, right? Your body temperature is maintained. We did talk about it. You have learned that in O levels. This topic was in the older syllabus. It's now excluded from this year's syllabus. Now, so same thing will happen when the temperature will increase above 30 degrees. So what will happen? Plants, they will start transpiring more and more to produce a cooling effect, to keep the plant body temperature lowered so that the enzyme can work perfectly. But eventually what will happen? Loss of water. And the plants are what? Mesophytes. So again, to prevent, you see, there are two problems at the same time. One, temperature is increasing. Second, the loss of water is also happening. So the plant will, you see, if there is enough, you see, if let's suppose it was a, potted plant, you are giving water and water, it's no issue for the plant. Enough water available. But if in the soil there is less water at that time, so it cannot lose any more water, otherwise the cells can become flaccid, it can wilt and eventually it can die as well, the plant. So even at that time when the temperature becomes higher, what they will do? They will close their stomata. Understand? And when the stomata they will be closed, water will be retained inside the plant body. Understand? So this is how they perform the process of homeostasis. Yes.
Doesn't a higher temperature cause more uptake of water?
More uptake? Yes, it will cause a more uptake of water, but we are talking about moderate quantity of water available in the soil. There is not enough water present in the soil to be lost. Right? So eventually what will happen? To preserve the water, whatever is present in the plant body and whatever is present in the soil, the plant will close down its stomata. Understand? Yes.
So we have these factors and all that the endocrine system for the external internal stimuli.
Yes.
So what is this system for plants? Like how do
Now,
we get in this?
they have hormones. We are going to talk about the plant hormones in the next chapter, inshallah. But we will talk about one hormone in this homeostasis topic as well, that is abscisic acid. Right?
So this change.
In the extreme conditions, like very high temperature or very low humidity, abscisic acid is released in the plants, and that helps in closing down the stomach. Alright? So they don't have nervous system like us, I told you in the beginning. But they do have hormonal system, and let me tell you, we have discussed this last year as well in the plant transport chapter. Right? That auxins or other hormones, they travel in the phloem. The hormones, they travel in phloem. Alright? Now, the next thing which we are going to discuss related to this topic, that is the opening and closing of stomata. But before that, we are going to discuss the structure of a stoma. Structure of a stoma. Now, I have drawn this diagram many times, so I am not going to draw it again. I am just going to draw the lower epidermis of this leaf. Only the lower epidermis. Remember last year I drew the diagram of the transverse section of this leaf many times. Right? Now, if I draw the lower epidermis, in the lower epidermis, we have these cells, right? Now, this over here is the lower epidermis. Now, remember one thing, guys. This opening over here and this opening over here, these openings are called as stomata. Another thing, stomata, it is a plural word. We have learned this before. Right? If I label just one of them, then it will be called stoma. This is singular. Right? Now, the cells that are present around these openings, these cells are called as guard cells. These cells are called what? Guard cells. Now, we are going to discuss the structure of the guard cells. Now, when the guard cells, they are completely turgid, at that time they are kidney bean shaped. Right? But when they are flaccid, they are of this shape. And when they are flaccid, at that time the stoma is closed. This is what? This is a closed stoma. Right? And these both cells, these are guard cells. Now, these guard cells, they have different thickness of the cell wall at different places. Now, this is the part of the cell wall of the guard cell which is facing inward towards the stoma. Which is facing inward towards the stoma. Which is also in contact with the air as well. This is in contact with the air, right? So these walls which are facing inward towards each other and they are also facing the air as well, these are thick cell walls. While the external cell walls on the outer side, they are what? Thin cell walls. Understand? Another important thing.
Sir,
Yes.
why is it thick?
Yes. There is extra deposition of cellulose and lignin on the inner side.
what's the use?
We are going to discuss
Okay.
that. Right now I am just telling the structure. Function we are going to discuss later. Alright everyone? So the inner walls, they are what? Thicker, and outer walls is what? Thinner. Another thing that they have foldings in their cell membranes. Now why there will be foldings in the cell membrane? To increase the surface area. To increase the surface area. The question is, why there is need to increase the surface area? Let me label this. This is the cell membrane. Increase the surface area for what reason? To increase the number of potassium ion channels, to increase the number of aquaporins, right? To increase the number of hydrogen ion pumps, etc. There are increase in number of chloride ion channels as well. Right? So I am writing etc. So main purpose is that to increase the surface area, so there are maximum number of potassium ion channels, maximum number of aquaporins, maximum number of hydrogen ion pumps are there, right? And chloride ion channels, etc. Calcium ion channels as well. Another thing which is very important, that these guard cells, they are smaller than mesophyll cells. Guard cells, they are smaller than mesophyll cells. Remember this. But the size of nucleus of guard cells and mesophyll cells is same. Let me mention over here. Size of nucleus of mesophyll cells and guard cells is same. It means that the nucleus will take up more space as compared to mesophyll cells. Now compare two things. Smaller than mesophyll cells, nucleus of same size as mesophyll cells. It means that inside the cell, the nucleus will take up what? More space. Understand? Another interesting fact, mesophyll cells have one large vacuole. Mesophyll cells have what? One large vacuole. They have many small vacuoles. They have small vacuoles. Next thing, that these guard cells, they have many chloroplasts and mitochondria. I am representing the chloroplast like this. And many mitochondria understand? So let me mention over here, have chloroplasts, but the chloroplasts of guard cells are a bit different than the mesophyll cells. The question is, what is the difference between the chloroplast of the mesophyll cells and the guard cells? The chloroplasts have less number of granule. The chloroplasts of guard cells have less number of granule. Understand? Next thing, that they have many mitochondria. Why the mitochondria are there? To produce energy, to produce the ATPs. Understand? Now this is what, this is a closed stoma. An open stoma would look like this. Now look here. This is what? This opening is called stoma. This opening is called stoma. And these are what? These are guard cells. Now we need to understand how the stoma open and close. How much time is left? I think so 5 minutes are left. Not enough time, but let's start this still. Now, for that purpose to understand, I'm drawing. Another thing which I need to explain before starting learning about that. Let's suppose that this is just one guard cell. Now, one more thing which I want to explain you, I will explain over here on this diagram. The cellulose fibers, they are arranged in this way. The cellulose fibers, they are arranged in this way on the guard cells. Now, what this is, you see, these cellulose fibers which are arranged in this circular way, and keep this in mind that outer wall is what? Thin. Inner wall is what? Thick. All of this is going to play an important role. This is the answer to your question. Circular arrangement of the cellulose fibrils, outer thin walls and inner thick walls. All of this is going to play a role in making the stoma to open like this. Understand? Now, this is one guard cell. And with the guard cell, over here, this is what? This is the epidermal cell. Lower epidermal cell, right? We know that, you see, they are arranged with this, right? So this is what? This is the lower epidermis. Now, we know that over here, this is the cell membrane, this is cell wall, right? Now over here, there is presence of hydrogen ion pump. What does this hydrogen ion pump do? It uses the energy from the ATP. An ATP molecule breaks down into what? ADP plus inorganic phosphate, and that energy is used to pump the hydrogen ions out of the guard cells. That energy is used to pump the hydrogen ions from the cytoplasm of the guard cells into the neighboring cells. And which cells are these? Lower epidermal cells. When the hydrogen ions, they will come out, inside there will be a decrease in positive charges. Hydrogen ions are positive charges, right? So when the hydrogen ion go out, there will be a decrease in positive charges over here. At that time, over here, the potassium ions, they are higher in concentration, right? So what will happen to compensate the loss of these positive charges? to compensate the loss of positive charge. These potassium ions also have positive charge, right? These potassium ions, they will start moving from the neighboring cells into the guard cells. Understand? Now, when the potassium ions will move from the epidermal cells into the guard cells, who can follow that?
Sodium.
Chloride. This is positive charge, right? So chloride ions can also follow them. Because this is positive charge, right? Potassium. Chloride is a negative charge. We'll follow that. So what has happened? That potassium ions and chloride ions, they will enter over here. They are diffusing. And which kind of diffusion is this? Facilitated diffusion. Which kind of diffusion is this? Facilitated diffusion.
So during reabsorption, does the same thing happen?
Sorry,
During
during
reabsorption, does the same thing happen?
reabsorption of what?
Of salts?
So distal.
This
Nephrons.
nephrons?
Yeah.
No, don't compare it with nephrons.
So if sodium is going to chloride,
But that, no, that is the secondary active transport. That is something else. This is not that. That is the secondary active transport. This is not that. And there are co-transporters. Here is no co-transporter. Got it? It's totally different procedure. Here, just the hydrogen ions are being pumped outside to compensate the positive charges. Positive charges are still coming in, right? And with that, the chloride ions will also come. Now, as these potassium ions and chloride ions are, what are these? These are actually solutes, right? So what is actually happening? The solutes concentration is increasing. Give me one more minute. Solutes concentration is increasing. Because of that, what will happen over here? The water potential will decrease. Got it? When the water potential will decrease, the water from the surrounding cells where the water potential is what? High, will start moving from higher water potential to lower water potential. From the surrounding epidermal cells into the guard cells. Understand? From here we'll continue tomorrow, inshallah.